we continue our lecture about lymphoid organs, and now we talk about lymph node, spleen, tonsils, and different other tissues. So as for the lymph node, first we have to say that lymph nodes are the part of lymphatic system which is represented by lymphatic capillaries and vessels. Those vessels, they carry lymph, which is almost the same as blood, just it is devoid of red blood cells, platelets. It contains only leukocytes and plasma. And what is lymph and how it is created? It is important in understanding of how this system works. Inside our tissues, blood capillaries, they bring not only oxygen but also through the wall of capillaries, liquid portion of blood percolates, giving rise to the tissue fluid. So our tissues, they are hydrated by the water coming from the blood vessels. And you know that inside our tissues there are collagen fibers, ground substance like hyaluronic acid, glycosaminoglycans, absorbing that water and retaining that water in our tissues. This water is needed to facilitate the movement of different cells, facilitate the diffusion and different metabolic processes, and exceeds of that water washes the cells and fibers from outside, and this dirty tissue fluid, exceeds of the dirty tissue fluid, is collected exactly by the lymphatic system. Why blood vessels can't absorb this dirty fluid? Because blood vessels should carry sterile blood. Blood should be pure. That's why we have lymphatic vessels parallel with the blood vessels, because lymphatic vessels, this is something like a sewage system in our organism. It absorbs and collects all dirty tissue fluids and transports it to the regional lymph nodes. So now we can comprehend the function of the lymph node. It collects dirty tissue fluid, which is lymph, and purifies it. So the main function of the lymph node is to purify the lymph from different antigens. So in case of inflammatory processes, for example in our throat, when we have a throat ache, bacteria cells, they are broken by regional immunocompetent cells, and through lymphatic vessels, those dead bacteria cells together with the dead immunocompetent cells, they are collected and brought to the lymph nodes. That's why usually we have enlarged lymph nodes during the infection process. And not only infections are collected, sometimes metastatic cells, you know that cancer, it sends metastasis, and they are also distributed through lymphatic vessels. That's why it is very important to understand how the system works. And I remind you that last lecture in the connective tissue proper we also discussed a special system which is called interstitium, parallel with the lymphatic vessels, lined with endothelium. We also have tubular-like structures lined with collagen fibers, also bringing tissue fluids and helping to deliver it to the lymph node. Okay, and now let's talk about lymph node and how it purifies the lymph. Besides, just after the lymph was purified, it returns back to the blood vessels through the thoracic duct. So purified lymph returns back to the bloodstream. Okay, so lymph nodes, how they are organized. First of all, they are represented by the stroma and parenchyma. Stroma, these are the three-dimensional scaffold, the skeleton of the organ. So stroma is represented by the reticular tissue. We discussed it last lesson in the topic connective tissue, and you remember that it is called reticula because it forms a meshwork, which is rete in Latin. Reticular because meshwork is formed, and it is represented by reticular cells and fibers. After that, parenchyma of the lymph node, as this is the lymphoid organ, is represented by immunocompetent cells, predominantly lymphocytes. And now let's discuss how it works. First of all, in the lymph node we distinguish three zones, three compartments. The outermost being cortex, which is dark and represented by lymphatic follicles. Here, lymphoid nodules of follicles. Please do not confuse lymph node as a whole and lymphatic nodule as a small node, or lymphatic follicle it is also called. And they are predominantly represented by B cells. After that, the zone number 2 is paracortex, or sometimes it is also called deep cortex. It is represented predominantly by T lymphocytes, or T cells. So while cortex is is populated by B cells, paracortex is predominantly populated by T cells. After that, we have the medulla, which is represented by medullary cords. Here they are of purple color, and there plasma cells, macrophages are located, and the function of every zone we are going to discuss later. So first, how lymphocytes do arrive to the lymph node, how B and T cells appear there. First of all, they could be brought with the lymph. As you can see, lymphatic vessels, they bring lymph to the node, and as we have said, lymph contains lymphocytes and blood plasma. Another way of how T cells arrive to the lymph node, this is through the high endothelial venules, which are located in the paracortex. So please, let's talk about, pay attention. Yeah, we want to highlight that these are very unusual vessels located in the paracortex, and they worth to be considered in a distant slide. So here they are, high endothelial venules. Why they are called so? Because usually endothelium is a simple squamous epithelium, but in this case it is high, tall, cuboidal, and that's why they have the name high endothelial venules. What's unusual with them? Because by passing through them, lymphocytes, when they pass through these venules, they attach to their walls, and they, through diapedesis, enters the lymph node. So high endothelial venules, this is the site of exit for lymphocytes from the bloodstream. And this event, this phenomenon is called homing, because something like lymphocytes, they return back home to the lymph node from the bloodstream. Okay, so lymphocytes, they appear in the lymph node through high endothelial venules. And as you remember, I remind you that lymphocytes, they recirculate in different lymph nodes, and they enter the lymph node. Here they are looking for the cognate antigen. If they do not find it, they return back to the bloodstream and exit in another lymph node or in another lymphoid organ looking for the cognate antigens. So lymphocytes, they usually migrate from one lymph node to another, and this is the normal process. Then the second function of these high endothelial venules, this is the fluid absorption with the help of the aquaporine 1. You remember that aqua, this is the water, porine, this means that this is the pores for water. Those pores, they facilitate the movement of water through the cell membrane. And aquaporine type 1 is characteristic for high endothelial venules, and this is exactly causes the lymph flow by fluid absorption, so they have double function. Okay, let's move on, and here we see the specimen of the lymph node, and let's find out what we can distinguish here. First of all, lymph node is covered with a connective tissue capsule. Last lesson we discussed connective tissue, and we said that it is present almost everywhere. This is the confirmation. So it is surrounded by the capsule. After that, this capsule, it sends trabeculi inside the organ. You can see here septa could be found. And after that, in the cortex, we can see lymphoid follicles. Here they are, represented predominantly by B cells. And then in the paracortex, in this region just under the cortex, we can find those high endothelial venules. Here they are, very prominent and could be very well distinguished. And these are medullary cords. Purple, they are stained in purple color because here lymphocytes are concentrated. And that's what we can find in the lymph node. And then let's summarize the functions of the lymph node. This is the filtration of the lymph-borne antigens. We have already said, yeah, it collects lymph nodes. They collect with the help of lymphatic vessels all the dirty tissue fluid from regional organs, and here purification of lymph takes place. And the second function is antigen-dependent differentiation of T and B cells. It means that lymph nodes they not only eliminate those antigens, yeah, they not only phagocytose them or just kill them, but they elaborate, they launch the adaptive immunity. Here they stimulate respective T and B cells to specifically reject those antigens, to produce specific antibodies, and that's what's very good shown in the video. I recommend you to watch. The hyperlink will be in the description below. Let's move on. And before discussing how it works, before general explanation of its function, we have to comprehend the system of sinuses inside the lymph node. So lymph comes to the lymph node, it arrives through the system of afferent incoming lymphatic vessels, and what's unusual is that lymphatic vessels, they come from different sides, because blood vessels, they usually enter and exit the organ through the hilum, through the same hilum. Yeah, in case of lymphatic vessels, they bring lymph to the periphery from different sides of the lymph node. Then lymphatic vessels, they bring lymph. Now inside a lymph node there is a system of cavities through which lymph is percolated, passing through the lymph node. And exactly by passing through this system of cavities, lymph becomes purified. So let's discuss them. First, by entering the lymph node, lymph appears in the subcapsular sinus. Sinus is something like a space. So subcapsular sinus is located between the capsule, as it's obvious from its name, and lymphatic follicles, between the cortex and capsule, subcapsular sinus. So here we have the space. After that, next sinus is trabecula or cortical sinus. It is located between the trabeculi and lymphatic follicles, this space. Here it is of yellow color. After the trabecular sinus, the next one is medullary sinus, between the medullary cords and trabeculi, or between medullary cords. So those spaces here in the medulla, we call them medullary sinuses. Eventually they fuse with the formation of hilar sinus, and through which efferent vessels bring lymph out of the organ. So lymph node receives lymph through afferent lymph vessels, and by percolating through the system of sinuses, it exits the organ through the efferent lymph vessels. Now we have to comprehend one more thing, is that system of spaces is not empty, but instead it is filled with a three-dimensional network represented by reticular endothelial cells. These cells are needed to, how it is written, it should be written here, to slow down lymph flow. So lymph passes through the lymph node very slowly, because sinuses are occupied by a meshwork of reticular endothelial cells. Why they are called so? Because by their region, they are the part of reticular tissue, they are connective tissue. By the function, they are endothelial, as they line spaces. That's why reticular endothelial. And this tissue is needed, to slow down the lymph flow, and also macrophages and dendritic cells are fixed in this meshwork by the one hand, and by another hand they are capturing antigens, passing by. So passing by these sinuses, passing through the system of sinuses, lymph is purified by macrophages fixed inside this reticular tissue. Okay, now let's discuss how this system works. Now we are ready. So, the system of events, how lymph node works. Antigens are brought with a lymph flow and are captured by macrophages present inside sinuses. That's what we have just discussed. So, inside sinuses, macrophages are awaiting, and they capture all the rubbish, all the dust particles arriving with a dead tissue fluid. After that, macrophages, by capturing those antigens, they migrate to the paracortex, which is the T-dependent zone, rich in T cells, and present those antigens with the help of MHC2 to T helpers. So what happens in the paracortex? Why do we have there so many T cells? Because after macrophages have captured antigens, they expose them on their surface with the help of MHC2, and they present those antigens to T cells. What do they do in the paracortex? Among majority of T cells, they are looking for those with their receptors cognate, with the receptors of high affinity to this particular antigen. So they are looking for the proper T cell to reject this particular antigen. And this is the essence. That's why we need to have a lot of T cells in the paracortex. Because in case of infection, we have exact antigens infecting us. And we need to find a proper T cell with its T cell receptor fit to this particular antigen. That's why macrophages do arrive to the paracortex. Okay, and that's why paracortex is populated by the T cells. Okay, in case the proper T lymphocyte is found, if T lymphocyte recognizes cognate antigen, it becomes active and migrates to the cortex. What does it mean it becomes active? If proper T cell is found, it undergoes multiplication processes, proliferates, it gives rise to many T cells with the same receptor against the same antigen. So, in case proper T cell increased in the number, it migrates to the cortex where B cells are located. And among the majority of B cells, only those become activated, which can recognize the same antigen as activated T cell. So you see the sequence of events. First, macrophage captures the antigen. After that, in the paracortex, it looks for the proper T cell, activating only proper T cell with its T cell receptor fit for this particular antigen. After that, this T cell increases in number, migrates to the cortex, where it looks for the proper B cell. And if proper B cell is found, it also increases in the number, and then it undergoes clonal expansion, it proliferates. And now let's look what happens further. Then activated B cell becomes a plasmablast, and there are two groups of plasmablasts. After they increase in the number, there are two ways. The first way, plasma cells immediately, just after the activation, immediately they go to the medullary cords, to the medulla of the lymph node, they descend to the medullary cord and start to produce immunoglobulin M. If you remember, those immunoglobulin M, they are of low affinity. They recognize antigen very weakly, but they could be produced immediately in a high amount, and they are pentameric in the structure. So they can neutralize antigen more or less effectively because of their large number, high number. The other group of plasmablasts, they remain in the cortex, and they create a lymphatic follicle or nodule. And that's exactly what's happening there, affinity maturation. So they start to improve their antibodies. They start to to modify it by increasing the number of mutations in its variable region. There is the process of somatic hypermutation, and in this way, the part of the antibody, which is destined to interact with the antigen, is changed. So, plasmablasts, which are left in the cortex, they are called centroblasts, and in the dark zone, they proliferate, starting to modify their antibodies, trying to make those antibodies more affinic to this particular antigen. After that, after they have changed their antibodies, they migrate to the light zone of the lymphatic follicle and test their antibodies. If affinity has increased, then such centrocytes do survive, return back to the dark zone, and continue to improve their antibodies. If by mistake, because mutations, this is the random process, mutagenesis, this is the random process, if the affinity was lost, or if the affinity has decreased, then such centrocytes they just die by apoptosis. So along 1 week, along next 7 to 10 days, some centroblasts they remain in the cortex and they continue to improve their antibodies, and after 7 to 10 days they produce antibodies of high affinity, which are called immunoglobulin G, and such plasma cells they can go to the medullary cords to produce more affinity antibodies. That's what is very important in understanding of how lymph nodes work. So just after activation of the B cells, they either go to the medullary cords to immediately produce low affinity antibodies, or they remain in the cortex. Another group remains in the cortex, starts to proliferate to improve their affinity, and in a week they really give rise to plasma cells producing more affinity antibodies, which are immunoglobulin G. Okay. And here the lymphatic follicle is shown in details, and here we can see the dark zone, light zone, and the mantle zone. So in the cortex of the lymph node, after antigen stimulation, after some B cells were activated by the proper antigen, what happens? Some B cells here, centroblasts, they divide and they increase the affinity of their antibodies to the antigen. After a set of divisions, they migrate to the so-called light zone. Here light zone is located. They become centrocytes and they test their antibodies. Here a special type of follicular dendritic cells are located. Their function is just to capture and to hold antigens on their surface. So they do not phagocytose, they do not digest antigens, but they simply hold, they retain antigens on their surface. So when centrocytes appear in the light zone, they try to test their antibodies, whether it has high affinity to this antigen, or maybe the affinity was lost. So to test this, they need to have an example of the antigen. And examples of those antigens are held by follicular dendritic cells. So the function of follicular dendritic cells is just to hold an example of antigen, exposing it to the centrocytes. So centrocytes with their improved antibodies, they test those antibodies. If the affinity to antigen has increased, such centrocytes survive and return back to the dark zone. Here they continue to improve their antibodies. If the affinity was lost or has decreased, such centrocytes they die through the help of apoptosis. Okay, this is the key event, what happens in the cortex of the lymph node. Affinity maturation through the somatic hypermutagenesis. And also there is a mantle zone. In the mantle zone B cells, there are resting B cells. They do not divide because they are not involved in the process of reaction against these particular antigens. So these are neutral B cells. Then, okay, that's what we had to know about the lymph node. Now we talk about the spleen. Spleen is another organ destined to filter our blood. As we have lymph nodes to purify lymph, the function of the spleen is to purify blood from different antigens. Okay, also we have kidneys, it purifies blood from toxins. Also we have liver, liver purifies blood from everything what was absorbed in our intestines. And we have spleen to purify blood from different antigens and dead or old senile blood cells. And first of all, we have to discuss the structure of the spleen. Spleen, again, is represented by the stroma, which is the three-dimensional scaffold of the organ, and the parenchyma, which is represented by blood cells. Stroma is made up of reticular tissue as well as in the lymph node. Parenchyma is represented not only by lymphocytes but also by red blood cells. That's why if in the lymph node we have only lymphocytes, in the spleen we have leukocytes and erythrocytes. That's why it has red pulp with erythrocytes in it and white pulp with leukocytes in it. White pulp is represented by the periarteriolar lymphoid sheets, perivascular, actually periarteriolar, there is a little mistake, and lymphoid follicles. So white pulp in the histological section looks like purple because lymphocytes, and they are stained with hematoxylin because they contain almost only nucleus. But if we look at a fresh specimen, if we cut the spleen before fixation and staining, really the red pulp would be of red color, dark brown, while white pulp would be white. So these are they are named after the color in a fresh organ, but in the specimen they are red or pink and purple instead of white. Okay, and before discussing the spleen, we have to discuss the system of its blood circulation, because it is impossible to understand how it works without imagining its blood vessels. So spleen is a very unusual organ in our body because it has open system of blood circulation. What does it mean? It means that blood just simply leak out of blood vessels. Let's discuss the system in detail. So splenic artery entering the organ gives rise to the trabecular arteries, which are inside trabeculi of the organ. And after that, central arteries, they enter the parenchyma of the organ, and they give small branches, side branches, but eventually they branch into penicillar arterioles. Eventually penicillar arterioles give rise to the sheeted capillaries. They are called sheeted because they are ensheeted with macrophages and leukocytes. That's what you have to know, and this is the most critical element of the splenic system of blood circulation, because exactly those capillaries, they become opened, and through those capillaries blood enters the parenchyma of the organ. So you can see here is the gap between arteries and veins. They do not communicate. That's why the system is not continuous but instead discontinuous, and that's what we call open system of blood circulation. Open system means that blood enters the organ through opened capillaries and then is absorbed or returned back to veins through blind-ended veins, which are called sinusoids. So in spleen we have blind-ended, highly permeable venous sinusoids, which controls the quality of blood cells and allow only those blood cells to come back which are of proper quality, which are qualitated enough. Okay, so in human spleen we have completely opened system, so all capillaries are opened, and that's what was confirmed by the set of articles and three-dimensional reconstructions. I'll give you the hyperlink in the description below, because in some textbooks we still can find the statement that spleen in humans has both opened and closed system of blood circulation. All the evidences says that really we have completely open system. Okay, and in this system it is important to understand how venous sinuses do control the parameters of erythrocytes to decide whether to return them back, whether not. We have open system of blood capillaries, and in this diagram we see that just after leaving capillaries, erythrocytes they get in contact with macrophages inside the spleen, and those macrophages, first of all, what they do, they clean the surfaces of erythrocytes, removing antigens from their surface, because you know that red blood cells, they also perform the function of capturing of immune complexes. The complexes of antigens and antibodies are fixed to erythrocyte through special receptors and are delivered to the spleen. So here in the spleen, macrophages, they clean the surface of erythrocyte, and after that, if erythrocyte has lost its negative charge, if it is distorted in shape, it is phagocytosed by local macrophages. So if this erythrocyte is damaged, it would be destroyed locally. Then next, if erythrocyte is spherical in shape, it would never enter the sinusoids because the gaps between endothelial cells in those sinusoids are longitudinal, are elongated. So only biconcave erythrocytes could squeeze between them. And if cell is spherical in shape, it is too large to enter those slits. Okay, so sinusoids, they perform the function of quality control if erythrocytes are not elastic enough or not flattened enough to squeeze between those slits. They would be entrapped in the spleen and eventually phagocytosed by macrophages. So erythrocytes are not absorbed by sinusoids if they are of low elasticity, if they are distorted in shape without negative charge, or not in compliance with the blood group in case of blood transfusion. So it is important to highlight that spleen performs very important function of the quality control. And macrophages, they engulf not only old red blood cells, but platelets as well, and of course all antigens possibly present in the bloodstream. And it is very important to note that in this way spleen prevents the spread of infection through the bloodstream. Then, now let's talk in details about the red pulp and the white pulp of the spleen. We have just considered the blood circulation. What about the red blood pulp? In the red pulp, here it predominates in the human spleen. Up to 80% is represented by the red pulp. And it is represented by reticular cells, erythrocytes, and macrophages. It is filled with the erythrocyte, that's why it is red. And there are lots of macrophages because they are eating old red blood cells and they participate in the recycling of their iron. Sometimes we might find the term splenicords or colds of Billroth, but this term is not very popular now, and it seems that it would disappear from the scientific literature because actually those spaces could be found only in the sections, but if we make a three-dimensional reconstruction, those cords do not exist actually. So this is the ancient term coming from the previous century. Okay, and now what about the white pulp? White pulp is represented by white blood cells, but they are distributed into zones. Like in our lymph node, we have B-dependent zone, the zone populated by B cells, zone occupied by T cells. In the same way, cells are distributed in our spleen. So let's comprehend it. First, inside the white pulp, we can find the lymphoid nodules, also called Malpighian corpuscles, and they are represented by the germinal center, which is rich in B cells. The same lymphoid follicle, the same as the lymphoid follicle or nodule in the lymph node. Then mantle zone surrounding it. Here we can see inactive B cells. That's why B cells not involved in the proliferation. They were not activated. They are not needed right now. And also it is called B cell corona. So the term is even more frequently found in the Western literature. After that, marginal zone could be found, and this is what we can find in the lymph node. Marginal zone is characteristic for the spleen, and it is rich in unusual marginal zone B cells, and their function we are going to discuss next slide. So marginal zone B cells to reside right here. What is unusual with this zone? Because exactly here antigens from the bloodstream arrive, because exactly here blood is pouring out of the opened capillaries. So this is the site where blood leak out, and it first comes in contact with local cells. And after that, periarteriolar zone, which is around the central arteriole. Notice that periarteriolar zone doesn't surround the whole follicle, but this is an additional zone attached to the lymphoid follicle. Here it is labeled with the number 4 in the diagram around the central arteries, arterioles. And perifollicular zone, it surrounds the whole white pulp. And perifollicular zone, this is the zone on the border between the white pulp and the red pulp. And now let's discuss how it works and how cells do interact one with another after then we return. So in the spleen, blood is pouring out of opened capillaries in the marginal zone and gets exposed to the local macrophages. In the same way as in the lymph node, lymph arrives to the sinusoids, to the sinuses here, blood brings antigens. Then macrophages, by capturing those blood-borne antigens, they migrate to the T cells dependent zone. Like in the lymph node, macrophages migrated to the paracortical zone. Here, activated macrophages with their antigens, they come to the periarteriolar zone, which is populated by T cells. So again, macrophages, they go to the T cell zone. And here antigen presentation takes place. So macrophages or dendritic cells, with the help of MHC2, they present those antigens to T helpers in the periarterial zone. So among the different diversity of those T cells, only those are activated which have their receptors cognate to the proper antigen. So macrophages are looking for those T cells with their receptors fit for this antigen. And just after they are found, they activate and they stimulate such T cells to divide, to amplify in number. And guess what? Those T lymphocytes, they migrate to the B cell dependent zone. Those activated T lymphocytes, they become active and migrate to the B cell zone, to the lymphatic follicle. So they go to the B cell corona and they try to find among inactive B cells those with their receptors fit for this antigen. And among all the B cells, only those become active which have their antibodies, their receptors fit for this antigen. In case of their activation, they become active, they divide, and they give rise through the process of clonal expansion, they give rise to a set of B cells with the same antibody specificity. So nothing difficult. Let's summarize this process. So what happens in the spleen? Blood is brought through opened capillaries to the organ. Then in the parenchyma of the organ, all blood-borne antigens are captured by local macrophages. Macrophages, let's return back. Macrophages, here we have marginal zone. Yeah, from the marginal zone, macrophages, they have captured antigens. They migrate to the T cell dependent zone, which is number 4. This is the periarteriolar zone. Here T cells are located. Here among different T cells, only those are activated with their T cell receptors fit for this antigen. Then such T cells, they proliferate, increase in the number, and they go to the B cell zone here. And among different B cells, they activate only those with their antibodies fit for this particular antigen. And after that, activated B cells, they have two fates. They either remain here inside the follicle and create the new germinal center, improving their antibodies and undergoing the process of affinity maturation, or another way, the second way, they immediately go to the border between the red and white pulp and start to produce immunoglobulins of low affinity, immunoglobulins M. So let's look here. The same diagram is in the lymph node. So after B cells get activated, they divide into two groups. One group immediately produces immunoglobulins M of low affinity. Another group remains in the B cell dependent zone, in the lymphoid follicles, and they undergo the process of affinity maturation through the somatic hypermutation. So they introduce mutations in the variable regions of their antibodies, and after that they test those antibodies, whether their affinity has increased. In this case, they survive and they continue to improve their antibodies. In case affinity was lost, such centrocytes, they die through apoptosis. Eventually, after 7 to 10 days, new plasma cells they really produce antibodies of high affinity, and that's why after 1 week of infection we usually can see the relief in the patient, and that's because new antibodies are produced, and that's why usually we have a flu for 7 to 10 days until affinity antibodies, highly affinity antibodies start to produce them. Okay, this is what happens in our spleen, and as you can see, the sequence of events is quite the same as in the lymph node, but lymph node deals with infection and antigens in our lymph, while spleen deals with infections inside our blood. That's why if spleen is removed, then patient suffers from infectious diseases more frequently, and eventually we have to discuss functions of a spleen. So first, immunologic filtration of our blood. It really purifies blood from blood-borne antigens. How blood could contain different bacteria. For example, in case we have acute inflammation in one organ, for example in lungs during pneumonia, pus could be formed and some blood vessels could be damaged, and through those damaged blood vessels infection might be absorbed by the bloodstream, and through the bloodstream it could be transported to different other organs. And to prevent that, we have spleen, because by passing through the spleen, this infection will be entrapped here and neutralized here. Then antigen dependent differentiation. So here antigens are not only eliminated, but here against these antigens specific immunity is launched. So spleen starts to produce specific antibodies to neutralize this infection in any other sites of the organism. So spleen is the very active site of antibody production as a result of activation of T and B cells. Then destruction of aged red blood cells and platelets. Of course, you know that old red blood cells are buried here, and together with the liver, spleen performs this function. But in liver, only very damaged red blood cells are destroyed. A spleen better deals with that function, because it can distinguish more precisely the signs of aging. That's why if spleen is removed, we can find many distorted red blood cells in the bloodstream, because liver can't cope 100% with this function. Okay, and also it recycles iron, recycles iron from hemoglobin, so iron couldn't be lost, and almost all iron is recycled and transported to the red bone marrow for the purposes of erythropoiesis. Notice that in some textbooks you are also given the information that spleen is used as a storage of blood. In humans it doesn't work, because a spleen is like a clenched fist. It is very low volume, and it has the capsule with very low smooth muscles in it, so it can't contract significantly. And many experiments were performed when people underwent highly exhausted physical exercises, and as a result it was the volume of spleen was measured, and it was found out that really the volume of spleen doesn't decrease significantly. So spleen can't inject a significant amount of blood to the blood circulation. If we compare with some animals, for example horses or some large cats like tigers, for example, when they are running very quickly, the horses, for example, could obtain up to 40% of red blood cells from their spleen. Up to 40% of all erythrocytes, they reside in the spleen, and they could be injected to the bloodstream when it actively when it runs actively, and that's how it increases its oxygen carrying capacity of the blood, by increasing the hematocrit. In humans it doesn't work, so under active physical exercises only a few red blood cells are injected. Okay, so never mind, let's go further. And after we have discussed the function of the spleen, of the lymph node, we have to say that there are many other lymphoid organs, and I want to highlight that up to 40%, not 40, majority of our lymphocytes, in our organism, they are associated with the digestive system. That's why you can imagine that lymph nodes and spleen, these are very important lymphoid organs, but our digestive system is very important for our immunity because it deals with many antigens coming with the food. And when we have the topic digestive system, we again return to our immunity and discuss its function as a part of immune system. But now we have to mention that in our oral cavity there is a lymphoid ring. It is located just at the entrance to the digestive and respiratory system, and it is represented by a set of tonsils, and you have to know them and to know the function. So first there is a pharyngeal tonsil, which is unpaired, and it is also called adenoids. It doesn't have crypts, and sometimes it is enlarged in children, preventing their nasal respiration through the nose. Then palatine tonsil, there are two tonsils, and they are located in the pharynx by each side, and sometimes they are removed, and it was a very frequent procedure in the last century. Nowadays I think it is not. Those tonsils, they often get inflamed, and it was thought that they should be removed to prevent the infection of the respiratory tract. But now we understand that they try to prevent the spread of infection further to the respiratory tract, and it is better not to remove them like that. Okay, let's investigate their structure. Here palatine tonsils are shown, and that's how they are organized. In the region of tonsils we can see a crypt. This is the invagination of the epithelium in the connective tissue, and crypt is lined with a special epithelium, which is stratified squamous non-keratinized. Now you know what does that mean. We have already had the topic epithelium. Notice that in this region, just under the epithelium, lymphoid follicles or nodules are located with B cells inside. These follicles, they contain B cells undergoing the affinity maturation, and they always interact with lymphocytes, which infiltrate the epithelium. So basement membrane is not very well developed here. And it is very important to take into account, because if this epithelium gives rise to cancer, to malignant tumor, its metastases are frequently distributed because of almost absent the basement membrane. Because here lymphocytes, they infiltrate the epithelium, and they frequently migrate through the basement membrane, almost destroying it. So what is the function of the tonsil? It monitors all the antigens entering our digestive system. They capture those antigens, leukocytes, they capture those antigens, migrate to the regional lymph nodes, and then return back to the mucosal envelope, and here they give rise to a set of B cells, producing antibodies or improving the affinity of their antibodies in form of follicles. So here we can see the first contact of our antigens with our immune system, and here the adaptive immunity is launched. After that, also in the same way, we have the lingual tonsil, which is unpaired at the root of the tongue. It is located at tubal tonsils. They are located around the eustachian tube opening, and laryngeal tonsil in the epiglottis. So they could be not developed in humans. But together, those tonsils, they represent a lymphoid ring, which is destined to capture antigens before they enter our digestive system. And here they start to launch the adaptive immunity against dangerous antigens, while they tolerate innocuous antigens. Okay, I think that's all what we had to consider this lecture, and at the end I want to give you an example from the USMLE database and to ask you to help me with the answer. I think you'll cope, despite the task is very complicated really, because it needs some knowledge from clinical departments, but I'll help you a little, giving you the hint. So in this task, 60-year-old boy is given, and he complains with fatigue and sore throat for past 3 days. At the same time, he doesn't have signs of acute respiratory infection. Yes, so his temperature is a little bit elevated, but on physical examination, his cervical lymph nodes are large, and also enlarged tonsils he has, but streptococcal antigen test is negative. But in peripheral blood smear, that's what's found, and that's what we have to concentrate our attention at. So such cells are found in the blood smear, and the question is, which of the following lymph node regions predominantly contains the abnormal cells seen in this patient's peripheral smear? So here different zones of lymph nodes are given, and the question is in which zone usually those cells are concentrated. And to answer this question, I need to say that this boy, he has Epstein-Barr virus infection. Epstein-Barr virus, it affects some immunocompetent cells, and as a result some another immunocompetent cells increase in number, and exactly those abnormal cells are found in the blood smear in this case. And to answer this question, you have to understand what exactly cells are here, what abnormal cells could be found in the blood smear in patients with Epstein-Barr virus, and in which zone of lymph node we could find them. Of course, you can't answer this question using only the material from the lecture. You should search and use some information from the internet, of course, but I think it will be useful for you to do that. Okay, hope you'll cope. So your answers please put in the comments below, and thank you for the attention. We'll see you next time. Goodbye.